Hey everyone, my name is Mayra,
And I'm Hannah.
and we're going to talk about Mendellian genetics.
Cool, but first, who is this Mendellian guy?
Well, Gregor Mendel was a monk who tried to understand what controls heredity in the world. Mendel lived during a time where chromosomes were yet to be discovered, but through his experiment, he was able to come up with two new concepts that are still used by scientists around the world today.
Gregor Mendel performed multiple experiments on pea plants because pea plants grow quickly, made a lot of offspring, and were capable of self-fertilization. Self-fertilization was essential to the experiment because Mendel needed to isolate certain genes in order to observe them.
Self-fertilization allows the pea plant to create offspring that only have one gene. For example, when a pea plant that produces only yellow seeds self-fertilizes, the offspring will only have yellow seeds.
This is important and we'll explain its significance later on. Meanwhile, let's talk about the two laws that Mendel created. The two laws he named the law of segregation and the law of independent assortment.
The law of segregation states that when gametes are formed, each gamete is given different allele. An allele is a form of a gene. For instance, the allele for color seeds of a pea plant could either be yellow peas or green peas. They are present on the same part of the DNA, so a gamete will only possess one version of the parent DNA. This is called being haploid. And when a parent cell makes gametes, it will undergo meiosis to create four separate haploid gametes. Because the chromosomes have separated, this is the law of segregation.
I love how you say, like, when a parent cell makes gametes, you say, like, and when a parent cell makes gametes. It's like kind of you're implying that, they're making babies. The law of independent assortment states that the genes carried on chromosomes that aren't homologous will randomly separate so that the inheritance of each gene will always be random, and they will not affect each other.
Now that we have the basics down, let's move on to Mendel's main discovery, which was done when he did a monohybrid cross, causing him to discover the existence of genes and alleles.
Okay, so let's use an example. First, Mendel crossed a yellow pea plant with a green pea plant. All of their offspring had yellow peas. So let's say that Mendel crossed two of the offspring that had yellow peas. And let's say that he discovered that 75%, or 3/4 of the offspring had yellow peas, while only 1/4 had green peas.
This helped Mendel discover alleles and certain properties of them. Each gene will have a dominant allele and a recessive allele. The dominant allele is superior to the recessive allele in that it will be expressed by the organism if the organism possesses both a dominant allele and a recessive allele.
And with that, we have to understand what homozygous and heterozygous means. Homozygous means that both alleles of the gene are identical. Heterozygous means that the two alleles of the gene are not identical.
Mendel didn't have the information or the technology to see which plant would get which alleles, so he bred the pea plant and waited to see what color their offsprings would be.
Let's use a hypothetical cross. You start with two pea plants, which were called the parent generation. Let's say that one of these plants carry a dominant gene for yellow peas and a recessive gene for green peas. This is represented graphically by a big Y, which represents the dominant yellow pea gene, and a little y, representing the recessive green pea gene. It crosses with a plant that only carries two dominant alleles for yellow peas.
Each plant carries two alleles because they receive one allele from each parent. In this example, we will be doing what is called a Punnett square. Punnett squares are used to determine the frequency or the probability of the offspring having a certain genotype, or the code, and a certain phenotype, or what is expressed.
So the female's genotype on the Punnett square usually goes vertically, and the male's genotype usually goes horizontally.
To fill out a Punnett square, you take one of the alleles that each parent gives and place it in the row that applies. We are assuming that the male gives 1/2 of his offspring the gamete that holds the big Y allele.
However, the other half will get the little Y gametes. And from the mother, because she has two big Y gametes, she will only give big Ys to her offspring.
If you notice, each bog has a big Y gene. Because the big Y allele is dominant, that means that every single offspring produced will express yellow peas. But one half of the offspring, the ones with a recessive gene, are called carriers. This is because they carry the recessive allele, but because they have a dominant allele, they cannot express the recessive.
This is what is called a monohybrid cross. It is called monohybrid because only one gene is being addressed. Let's look at another one. Let's say that this cross is between two carriers of the green P allele. So they are both big Y, little y.
If you see, 1/4 of the offspring will inherit gametes that both contain the yellow allele, or the dominant. I gotta do that again. Thanks, Santa.
Oh my god. Just do it.
If you see, 1/4 of the offspring will inherit gametes that both contain the yellow allele, or dominant. There are 2 boxes where the offspring are big Y, little y, like their parents. This means they are also carriers of the green P allele. However, the last 1/4 of the offspring inherited both the little y's from their parents. They will express green P.
Because we have 1/2 of the offspring as carriers, 1/4 is yellow non-carriers, and 1/4 is green non-carriers, we can set up our proportion for this graph, called the 1:2:1 ratio.
So 1/4 of the offspring will have 2 dominant alleles. This is called being homozygous dominant. 1/2 of the offspring will be carriers of green peas, but express the yellow peas. This is called heterozygous.
Lastly, the ones that express green peas are called homozygous recessive.
Let's use another example. Let's do a crossing using 2 genes that aren't related in any way. For example, pea color and plant size.
As we said before, yellow peas are dominant while green peas are recessive. And short plants are dominant, represented by a big S, and the tall plants are recessive, represented by the little s.
Both parents are heterozygous in both pea color and plant length.
Say that again 3 times fast.
I can't.
Do it.
I can't. Both parents are heterozygous in both pea color and plant length.
How much would woodchuck if woodchuck could chuck wood? How much would woodchuck if woodchuck could chuck wood? How much would woodchuck if woodchuck could chuck wood?
Red leather, yellow leather, red leather, yellow
I can't
leather,
do that.
red leather, yellow
Toy
leather.
boat. Lemon, lime. Okay, let's get back on topic.
Sorry about that,
Okay,
guys.
genetics. All right.
What were we talking about?
Cool.
Let's set up a Punnett square. Both parents are heterozygous in both pea color and plant length.
That was really articulate.
I think you.
All right. This is how you do what is called a dihybrid cross, meaning that 2 alleles are getting crossed. On the female column, you will write big Y, big S, big Y, little s, big S, little y, little s. These are the various gametes that the female can give to her offspring. Because the male has the same genotype, you will write that on the horizontal male line as well.
Set up the Punnett square bigger than a monohybrid cross because you have to write 4 letters in each box.
After you label the 2 sides, pair up the alleles. Do it for all the letters and fill up all the spaces in the Punnett square. Now, to make sure that the cross you have is the dihybrid cross, count all the alleles that are similar. For example, count up all the tall yellow pea plants, all the tall green pea plants, all the short yellow pea plants, and all the short green pea plants. In this Punnett square, we circled the yellow short pea plants with a yellow marker, and we circled the green short pea plants with a green marker, and we circled the green tall pea plants with a black marker. And as you can see, the ratio of this phenotype is 9:3:3:1. And that is the ratio of a dihybrid cross.
Cool. So the 9 is the short yellow pea plant because they are dominant. Both of the 3s are either tall yellow plants or short green plants. Lastly, the 1 is a tall green plant because they are the least common, having both recessive genes. So we covered monohybrid cross and dihybrid cross. Hannah, are we missing anything?
Oh, wait, we have to do the sex-linked crosses.
Oh, yeah.
Or yeah.
So So what are sex-linked genes?
So articulate. Sex-linked genes are certain genes that are passed down to offspring via sex chromosomes. For humans, genes for color blindness, placement of sweat glands, and certain types of baldness are sex-linked and are located on the X and Y chromosomes. Usually the X.
That's freaky.
Yeah.
Anyway, so how do we determine which offspring gets which genes?
As always, we reference the magical Punnett square.
Now, instead of writing a single letter to represent the allele, we have to write the sex chromosomes as well. Let's use the example for red-green color blindness. Color blindness is on the X chromosome. The dominant gene is the ability to see two colors. So we will represent this gene with a big A. The recessive gene is not having the ability to see the two colors. So we will represent this with a little a.
So let's say that the mother is heterozygous for color blindness, but the father doesn't have it. If we fill out the Punnett square, we see that if the parents have a female, then there is a 1/2 chance she will be a carrier and a 1/2 chance she won't. However, if they have a boy, there is a 1/2 chance that he will be colorblind because males only receive one X chromosome. This is why for certain genes, males are usually more susceptible to them, like hemophilia and stuff like that.
Oh, and that's why the chance of having a boy or a girl is always 50-50.
I mean, there are a bunch of other crosses out there, like linked crosses and stuff, but we aren't going to get into that. We don't want to confuse them.
Yes. Okay, yeah, let's not confuse them. Thanks for watching.
Bye!